I also want to thank the Faculty of Arts and Science for organizing the series and for inviting me to speak, because that is a really, really big honor, and that's humbling too. So I really need to try now to do my best, because it's very special to be here today. We have so many professors, and I am really grateful for the opportunity. I also want to thank right away Raj from the Alberta Epigenetics Network. We've worked together for many years. It is great to have this partnership. It's great to have the sponsorships. It's so nice to have food here and everything, right? So this is really important, because we can't do science and our work without sponsors. And I want to thank Alberta Epigenetics Network in particular. But having said that, what is the most important today? I really need to thank you for being here. This is, oh wow. I mean, look, the room is packed, and you all came to hear about RNA, and I'm not sure. Let's be honest. Who of you has heard about RNA before reading this title? Awesome. That is more than I thought. But now, like really, be honest. Who of you thinks you know something about RNA? That's still pretty good. Thank you. Those are all, half of those are the people I know from the university, right? I'm really thanking all the others for being here, because you are brave. And obviously, you trust me. So I'll see. I'll try to do my best, so that in the end, you not only know what RNA is, but hopefully you appreciate it, and you'll see that it does matter. So with that, we've covered the title. I'll start, and in one slide, kind of tell you in a nutshell what RNA is. And then we'll go into a little more details, and I'll also tell you what to expect tonight. So this is RNA. It looks pretty if you blow it up. In reality, it's so small you can't see it. It's a biomolecule. It's found in all our cells, in humans and animals and plants and bacteria, everywhere. It's in every cell. RNA sounds a little bit like DNA, and that is for a good reason. DNA is the genetic material in all cells, and RNA is a copy of DNA. And that sounds boring. And that's why for many years researchers didn't care about RNA, because it's just a copy, right? And then you throw it out when you don't need it anymore. Well, we'll see today that the copy matters a lot. And to just give you one argument right away, in a human body, obviously all cells have the same genetic information. Yet a brain is very different from a liver. Why? Well, because we use the genetic information differently. And how do you see that? You see it by which pieces get copied into RNA. The RNA in a liver cell is dramatically different from the RNA in a brain cell. And that's good for us, right? I won't dive into all the details tonight. That's a little bit too much. But let's take that as a first hint that RNA does matter. So what can you expect tonight? This is the take-home message. So if you need to leave, take that with you. Right? That's okay. If you get a call because your kid is sick, that's okay. I know those things. I've done it myself. Here you go. RNA is important. Why? It's important for medicine. I alluded to that in my talk. I'll have at least one example later that it's important for agriculture, and I hint at a couple more, because it's everywhere. It's important for so many areas. Sometimes that dilutes our message. That's the problem with the RNA researchers. They like to talk about everything. So I'll try to limit myself and focus on medicine today and basic research. So why is RNA important? I'll give you two main reasons with a couple examples. The first one is RNA is a target for novel therapeutics, for new drugs to cure diseases. And RNA is a very important target, and maybe a little bit easier to target than the traditional targets, which have been proteins. But even more fascinating for me, and for the moment you need to trust me, RNA can also be a tool. It can be the new therapy itself. You can use RNA as a drug. And I have a wonderful story that I'll share with you in the end. That's not from my lab, but that just is really fascinating to me, and I'll tell you that at the very end. And of course, as Matthew already mentioned, we are very excited about RNA and uLethbridge because we have an RNA Institute, and uLethbridge is, believe it or not, the Canadian Center in RNA research. We really lead the field. There's another group out in Quebec which is doing related work, but uLethbridge is kind of central, right? So you should know why, because of RNA. So really quick, what to expect today. I mapped it out in the title. We'll start with the beginnings of life. A little bit crudely, but we'll start at the beginning, right? And RNA matters. And I just want to lay the ground there and explain you a little bit what RNA is. So I felt that's a good starting point. Then I want to introduce the Alberta RNA Research and Training Institute a little bit. And I'll use that to also show you the breadth of RNA research and the breadth of where RNA matters, with a couple quick examples. I won't dive into details. If you really like that part, let the faculty know, and maybe some of those people can give a big public professional series next year, right? Not my job. Some of them are here too. You can ask them afterwards. Then I guess I should talk a little bit about my research, so I try my best to explain what that modification of RNA means and why it matters. And we'll end with two of the most exciting stories in the field of RNA research, where it really matters and where you can use RNA to, in the end, cure diseases. Okay, the beginnings of life. That's a big topic I set myself up, so we'll skip a few things. So let's keep it simple. Yes, this is my daughter. And, you know, like we are all, the children do the same. They ask, right, where am I coming from? And what was first? In her case, she asked about the apple and the apple tree. Others ask about the chicken and the egg, right? It's the question which keeps us interested. And so you try your best to explain a few things. She's grown a bit older by now. And we know a bit about evolution. I'm not an evolutionary biologist, but you try. And basically, keeping things really simple, we can kind of explain how organisms evolved from each other and with each other. And we can trace it back, and there's the idea that makes sense. There's good evidence that there was a common, universal single cell as the ancestor, and then it became more complex and multicellular organisms and so on. So we can kind of trace it back to that single cell. And my daughter doesn't ask me about the single cell because she has no idea what that is, right? So I've already taken you on a roller coaster. But if you get down to that level, even a single cell is pretty complex. And I, as a biochemist, I'm a little bit overwhelmed by a single cell because I know what's all in there, and that's kind of too much. There's DNA in there, the genetic information, obviously RNA, there are lots of proteins, there are lipids or fats and sugars and whatnot. So we still have a problem. We still have a chicken and egg problem of, well, where's that cell coming from? What was first? I leave that question hanging here a little bit and tell you a little bit more about DNA, RNA, and proteins, because we kind of need this today, and then we'll see if we can answer that question, or try at least. So, DNA, RNA, protein. That's the biochemistry take-home message here. So it's a little bit of teaching, sorry. DNA is the genome in our cells. That's the information, and it's double-stranded structure. It looks like that one on the top. And it gets copied into RNA, right? We talked about that already. RNA is the copy. And then the information in RNA is used to make proteins in a cell. And proteins are really the executors. That's what we know since 50, 60 years, that they execute a lot of functions. They make energy, they use energy, they make your muscles smooth, and all those kind of things. So most importantly, if we think about a cell dividing and making a new cell with the same genetic information, it's the proteins that copy, for example, the DNA into an identical new version. Very important function. So that's the first step. It's proteins that copy the DNA into RNA. That's up there. Then interesting, the step to go from RNA to protein. Proteins play a role, but RNA as well. So in that step, RNA and proteins work together to actually make proteins, right? And that's all the functions that you have in a cell. So many things need to be made, and that all happens with DNA and RNA. Oh, sorry. All those functions occur with RNA and proteins, not DNA. So that's complicated, right? That's why I kind of said we have a bit of a chicken and egg problem, because how did that arise in the first cell? That's already a little bit too much. Let's still look a little bit further at RNA. So it's a copy of DNA. This picture illustrates how it's copied. You just take the DNA, which is the famous double helix, open it up a little bit so that you can get at the sequence in the middle, and then you can copy it one at a time into RNA. Because DNA and RNA, and I won't go in many details, you have the same building blocks that are complementary. 4 building blocks, and 2 of them always match. If you've had high school science, you may remember this. There's a G matches a C, and an A matches a T. That's the only slide where I have A, T's, G's, and C's. From here on, we'll skip that. We'll just talk about building blocks. Don't want to make it too complicated. But they match. And that's how the sequence can be copied one into the other and back. And RNA has basically this very similar building blocks to DNA. Yet it differs, right? So you see a theme here. Talking about DNA, RNA, DNA, RNA. Similar. I claim RNA is more interesting, better. Well, that's not a good term to use in science. So what's the difference? So let's compare that, DNA and RNA. DNA on the left side is this double helix. It's kind of a famous picture. Watson and Crick found that in the 50s. RNA on the right looks more complex. What you see there is DNA is always 2 strands. RNA is a single one. And if you want to have a challenge, trace it. Start at the beginning and then try. You will see that you can go all the way around and back. It's one strand that winds around itself. So that's complicated. I think it's pretty. It's the basis for RNA being able to execute functions because it has a distinct shape. It's not just a helix. It always looks different. I'll show you some other examples. I know we have a couple chemists in the room. So for those of you, so what's the chemical difference? You see those blue circles, and there's a little red dot in the blue circle. You can barely see it. That's like one of those find the difference pictures. That's the difference. It's 1 oxygen atom, more in RNA than in DNA. That's like nothing from a chemical perspective. Like, it's so small. Yet this teeny tiny difference makes a big difference in how the overall structure looks like. And it makes an even bigger difference in the function of RNA compared to DNA. DNA stores the information. So I'll soon show you RNA is doing everything. Okay, that was the hardcore part. From now on, things get easier. Okay, thank you for bearing with me at the beginning. I kind of needed to get this out of the system so that we have a minimal common agreement of what RNA is. So let's get back to the simple. The problem, the chicken egg problem of where was this first cell coming from? And how did that happen with DNA and RNA and proteins? Which of those molecules was first? Right? So it's a chicken egg problem with 3 components. Wow, that's mind-boggling. So can we make this simpler? DNA, RNA, protein. That's every cell has that. Every bacterium, every human cell, every animal. Is there any way of making it simpler? Well, we can make it simpler because we can take both the DNA away and the proteins. RNA stores information. It's a copy of DNA. And that means you don't necessarily need DNA. You can have the information just in RNA. And you can copy RNA into RNA. We know that. That works pretty well. So DNA might have come later because it's more stable. But in the very beginning, RNA was probably good enough. But what about proteins? Like, who would copy RNA into RNA? Usually that's what proteins are doing. And that was a huge breakthrough about 15 years ago. And we found out that RNA can do that. RNA can copy itself without any protein, just RNA on its own. And RNA can do many more functions that I won't even dive into. RNA can be like a protein. It can catalyze reactions. So RNA has 2 functions. It can store information, and it can execute functions. And so the idea is that maybe, we have no proof of this, it's just an hypothesis, maybe at the beginning of life, there was an RNA world that consisted only of RNA. You needed 1 molecule that could do it all. It could store information, it could replicate itself. And then later, DNA was invented because it's more stable, protein were invented because they're more versatile, but at the very beginning, as a minimalistic version, there was probably, maybe, RNA. So RNA matters. Well, that's been a long time ago, right? That's the beginnings of life. I will get to why RNA matters today, but this just sets the ground. And by the way, if I completely lose you, or if you get really excited about something, ask me in between. I'm not used to teaching and talking the entire time myself. My students ask me questions, and I'll be happy to answer your questions in between. because we're actually at a crossroad anyway. We're switching topics now, so feel free to jump in. Yes? What is RNA for?
I mean, it copies itself, it executes, but why do we need it? What does it do for us?
I think the easiest for me is to go back to the answer I gave you on the first slide, where I said the RNA in your liver cell is different from your brain. So it determines which information gets used when to execute a function. And if you think about, we're getting sick, just having a flu, our body reacts. And all those reactions, that means we use different information. All the information is stored in the DNA, but we decide, oh, now we need this piece, or now we need that piece. And that gets executed through RNA. Does that give a good enough answer for now?
Yep.
Thank you. More questions? I'll give you a couple more examples now, because I'm about to introduce the Alberta RNA Research and Training Institute and the breadth of RNA in Lethbridge. There's even more out there. So here are the professors. We are 8 by now. We started in 2011. But what really matters is not those 8 people, right? But what really matters are the 85 trainees on the ground. And I have a picture of those, but I figured if I show that, you wouldn't recognize anybody. But they are really important. Some of them are here. Of all the young people in the room that I see, you have probably a 70% chance of that being one of our students. So talk to them. Really, do me the favor, talk to them. They know what really happens. And nothing will happen without our students. I mean, we are here for the students and not the other way around, right? So really important to me to get that statement out. So let's see a couple examples of what the groups working together with these professors and all these students are doing. So first example here is Dr. Trusha Patel. He joined us last year. He's here. Hi, Trusha, in the back. Yes, good idea. And what Trusha is studying is RNA and viruses. When I told you that in the RNA world, we may have had only RNA because RNA can store information, one very important argument supporting this idea is that nowadays, many viruses don't have DNA. They only have RNA. And RNA stores the information, the genetic information of the viruses. One example here is an adenovirus causing cold-like symptoms. Trusha studies how the RNA looks like. You see a beautiful example here, how the RNA interacts with the human proteins in our body, what happens during a viral infection, and how can we maybe fight that? How can we disrupt what happens? He also studies other viruses. You may say, who cares about a cold, we'll survive it. But other viruses that have an RNA genome as Ebola, Zika, hepatitis C, West Nile, measles, right? And all those are really important topics. So understanding RNA directly relates to understanding viral diseases, and understanding is the first step to developing a cure. I'll leave it there. I'll just go quickly through a couple other examples. So if you want to know more, Trusha is over there. Next one is Neha Sakho. I don't think he's, oh, there you are. Awesome, thank you, Neha. So Neha's lab is studying the regulation of how proteins are synthesized from the messenger RNA. So that's a direct example to the question I got of why RNA matters, because depending on which state a cell is in, you need to make different proteins. And some proteins are really important when a cell gets into a crisis. What does that mean? When a cell gets into a crisis, a cell can decide to undergo suicide, which is a good thing, because a crisis means that the other option is that the cell becomes a tumor cell, a cancer cell, right? And that is about the worst scenario. So lucky us, there are mechanisms in cells that, when they go into a crisis, to commit suicide. And Trusha studies how RNA regulates that. And so it's directly related to cancer research, what he's doing. And this is an example of a really complex RNA that is being researched in his lab to understand how it works. And then, once we know how it works, we can hopefully learn how to influence it. Let's keep going. Switch gears a little bit. Dr. Macrocell could not join us today. He's actually the longest RNA researcher on campus, so maybe you've met him somewhere. He's been here quite a while. He's a physical chemist from training, so he goes about things to really get at the principles behind it. He uses a lot of modeling to see what RNA is doing, and that gives us really deep insight. We need this. We all collaborate with him because it allows us to get a deep level of understanding. And the better we understanding, the better we can build upon it. And in particular, he models the process of gene expression, how DNA is transcribed into RNA and how that is regulated and how it is changing and how fast it's changing, right? Some things in our body are so fast. It's so amazing how quick our body can react. I always use the bad example. I'm German, right? So when I teach in my beginning, I was like, oh, yes, and there's the bear, and what do? You run. And my students told me, no, you don't do this, Ute. So that's the worst example ever. But my point was, you react, you're fast, you do something. Now, not with the bear. I learned that by now. So I need to find a better example. And here's somebody you should know. Stacy Wetmore. Stacy's there. Hey, Stacy. Isn't this fantastic that all my colleagues are coming out to support me? I'll come back to that in the end, but it really means a lot to me. Stacy is my role model. She gave a public professor talk in March, and you really missed something if you weren't there. She talked a lot about her fantastic research in DNA repair. She also mentioned that she's looking into RNA repair, how to fix RNA and RNA salvage, how a cell can recycle RNA. And that is, for example, a protein shown here where she studies how it's happening. And this is a very promising target for new antimicrobials, because bacteria really rely on this enzyme. And once Stacy understands how it works, and she's really close to that, she will find out right away how to inhibit it. And that might be an antibiotic for the future, right? So very important work there. And then I figured I didn't know how much time I have, and I know I like to rant, so I did a shortcut here. Dr. Athanasios Soboilis is here. Thank you. Dr. H.J. Whedon is here, our director. And I think Tony Russell couldn't make it, but I know he's here with us in spirit. 3 other people who really are core members of our team studying how the entire RNA in a cell is expressed and regulated in cancer and under stress. That's Aisen's work. Studying how ribosomes or protein factories work in bacteria and how they are targeted by antibiotic. That's Dr. H.J. Whedon. And Dr. Tony Russell is looking into the evolution of RNA in simple organisms. And now you might say, who cares? Well, organisms like Giardia that are causing diseases. And so that just shows you really the breadth of research that happens in RNA. But hopefully it also shows you some examples, just glancing on the surface, how RNA is related to cancer, to viral diseases, to antibiotic targets, so that's bacterial infections, and many other diseases. I'll show you a couple more examples. I need a moment to breathe. Do you have a question? Well, I guess if you have a question about this part, look at those people after, right? They can explain better than I can anyway. Okay, I think we are ready to dive in. What is going on in my lab? I have some experts sitting over there. Some of my students are here, so if you think they can explain better, I think so too. Ask them. I'll give you just the starting points. So as Matthew said, my lab is studying RNA modification. So what does that mean? I will start with pretty basic research here, and then I'll mention how that can apply to biomedical research. RNA is made of 4 building blocks. That keeps it very simple, and that's very important because that means it's easier to understand RNA. 4 is a number even a stupid person like me can understand. It means it's easy to copy. We know how it binds to DNA. We know how RNA binds to itself because we can just match them up. But 4 is also kind of not a lot, right? Maybe that's why proteins were invented, because they have 20 building blocks, so they can do much more fascinating things. Well, once proteins were there, proteins evolved that can modify RNA, meaning an RNA that has originally 4 building blocks gets altered so that there are over 100 different building blocks. And this, what I'm showing here, is just a simple example. It's called a transfer RNA. It's involved in protein synthesis. And on the left side, you just see the standard representation, but on the right, I highlight all the new modified building blocks in there, and there are 10 in this small little RNA. So more than 10% of its building blocks are actually changed. And sometimes the change is really subtle. Like this is another search the difference picture. As the typical building block is a U or uridine, and it gets changed in something that is so similar that we even call it the pseudouridine. And you would have to look really far to find that red little N that is the difference. So a really small chemical difference, but again, it stabilizes RNA. It makes a big difference. It's the most abundant modification. And one of the key questions that keep me up at night is, why? Why? This teeny little thing makes such a big difference. What is going on? And we have a lot of work ahead. So I can only give you a couple glimpses here today because I don't know yet, right? I hope I still have, I don't know, 25, 30 years or so to find out. So let me tell you one story that we published last year. My really great PhD student, Laura, worked on that for many years. And it's kind of not fair to her to summarize it in one slide without showing any of her data. But I think that's what today is about, right? We'll focus on the essentials and not all those details. So RNA modification is the chemical change, making a new building block. Why does it matter? We've proven last year that two things happen at the same time. The RNA gets modified, but it also gets folded into this correct three-dimensional shape. And that is very important. RNA needs this shape to function. Without that shape, it's useless. And so two things happen at the same time. And how does that work? Well, I brought you a little example. This. So I'm not good at crafts. So when I make these things, I always make a mistake, right? Something happens, bend it in the wrong direction or whatever. The same can happen with RNA. RNA can be folded into the wrong shape accidentally. And that's not good because that means this RNA is useless for the cell and the cell wasted energy making it and can potentially do harm in the cell. So now what happens when a modification enzyme or protein comes to modify this RNA? Well, first of all, the enzyme needs to get access to that green stuff in the RNA, those 3 green building blocks. So you want to get access to something, you take it apart, you open it up, right? That's what I'm showing at the bottom. Those green things, building blocks, they move out so that you can get at it. And then that's the first step. It's an unfolding step, right? I'm kind of trying to illustrate that here with my little paper too. You unfold so you have to gain access. Then modification can occur. Very interestingly and a little bit disturbing to me, we have shown that you could even skip that step. It's not that important, apparently. And what happens afterwards? Once the RNA is modified, you fold it back, and it has a good chance of being folded back correctly. And when it's folded correctly, you get a pretty 3-dimensional shape back. And that is not just pretty, because that doesn't matter in science, but it's functional. This RNA can now work in the cell, whereas the original RNA that was misfolded couldn't work. So the process of modifying RNA at the same time folds it correctly. So it's like 2 in 1, right? Like when you buy shampoo and conditioner in one. 2 in 1. It's very efficient. Nature is very efficient. And this is an example how it is very efficient. And I think it took us so long to find this because we scientists are sometimes simple. We think 1 in 1, right? Oh, we know this. It's an RNA modification enzyme. Nobody thought about that. It can also fold RNA at the same time. And this opens many new avenues for us. Just want to give you one example. A colleague of mine from China, he visited in August, he has found in patients a mutation. And the patients he's studying, they have impaired hearing up to the point where they are deaf. And they have a mutation in the RNA, a small mutation. And it's clear from the mutation that this inhibits modification. And that's what he knew. But when we started talking and he learned about our research, we realized what might be going on in the patients is not only that there's a mutation and it prevents modification, but likely that also prevents correct folding of the RNA. And that can have dramatic consequences. So maybe in this case, the hearing impairment comes from a problem in RNA folding. And we know of many other diseases that result from changes and mutations in the modification enzymes. And again, if those are altered, modification can't happen, and that means maybe folding can't happen. And so possibly we found a cause for those diseases. We don't know this yet. We still need to prove it. It's a lot of work. But that can affect neurological diseases, muscular diseases, cancer, to just name the few most important. So really, we think that we found something that is really worthwhile to investigate. It will take us another 10 to 20 years. Research is slow. Ask my students. It's painstaking to do the experiments, but I think it's also exciting because it does matter in the end. And having a new starting point, having a new hypothesis is really exciting. So what I'm doing is not medical research. I'm not claiming that. I'm not curing any patients. Sorry to disappoint you. But I hope that the type of basic research that we do matters in the long run. And I will show you an example at the end of research that others did where this was the case. Basic research leading to a drug. So RNA modification, RNA folding. Let's go through one more example from my lab. We also study, and it's related, how cells make their protein factories. Those are called ribosomes. I couldn't quite skip that word all the time. Sorry for mentioning it. Protein factories are obviously important in the cell because they make all proteins. And as I mentioned earlier on, protein factories are composed of RNA and protein. And in fact, RNA sits at the heart of them. So RNA is the most important component. That's why maybe in the RNA world, RNA was able to make proteins, even without proteins. So that was back the RNA world. What about today? We really like to understand how these big, what we call machines, are made out of RNA and protein. They're machines, but as Matthew said, they're so tiny you can't see them. You need to put a lot of RNA and protein together. We don't understand how that works yet. It's like me looking at a car and trying to understand how it works. There's no way I'll figure that out, right? And even me looking at a ribosome, trying to understand how those pieces were put together, it is mind-boggling. So we decided to start at the very beginning when things are still simple. As you can see, I like simple things because maybe I'm not smart enough for the others. The very beginning is that you have RNA, ribosomal RNA, and guess what? It gets modified. And that's what we are studying. How does it get modified? And then we also want to study how it gets folded, right? That's the theme here. But why do we do this? On my next slide, eventually, we'll see that there's a machinery in this case that does the modification. That's a little bit more complicated than the one I was talking about before. And most importantly, when there are mutations in the proteins during the modifications in ribosomal RNA, that gives rise to a genetic disease. So an inherited disease that is called dyskeratosis congenita. Who has heard about dyskeratosis congenita? Yeah, see? That worked. Those are the people working with me. Now you know them. Tricked you. So those are the experts. Well, you have not heard about it because it's a very, very rare disease. Okay. What does that mean? The disease is so rare that no pharmaceutical company has interest studying it. Because even if they would find a therapeutics for that disease, they could maybe sell thousands per year on the whole planet, and they wouldn't make any money. So this is an example where the companies can't do that research. They don't have the money, and they can't afford it. It's not commercially viable. But I think that's the reason why we need basic research at the university that's funded by governments, run by students, great students, who get trained along the way, and hopefully we can make some progress for those patients. And in fact, Canada has now a network of researchers studying rare diseases. Neil Thacker is also involved in that. And I think it's a really fascinating way to finally make progress on some of those problems that otherwise don't get tackled at all. So this is rare disease, and we know what mutations are there. So because basically we know there are mutations, and they result in changes in the protein that makes RNA modification. And then we know the patients are sick. They have bone marrow failure. It's a premature aging disease. Usually they die before age of 30. They have issues with any replicating tissue that grows fast, like nails, skin. So they have a very particular way they look like, but the most devastating is the bone marrow failure, because that can lead to death. They also have a higher risk of developing cancer. But why, right? I mean, how is that related from going from the change in the gene to all those symptoms? And we can try to, fix the symptoms, but usually that only helps temporarily with those patients. I mean, bone marrow transplantations are about the best you can do at the moment, but it's not really helping. So what we try to understand is what happens on a molecular level. What happens on how the RNA gets modified? What happens during ribosome formation, during forming the protein factories? Are there ways that we can maybe find a drug that helps at that step and whatever is wrong there in the function, right? But we are only at the very beginning. We don't know it yet. So what you also need is a lot of patience and a lot of good hope conducting this research. So ribosomes make proteins. And that is very, very important in cancer cells. What do cancer cells do? The tumor grows really rapidly, right? I mean, that's the problem. And unfortunately, we all know somebody who has had cancer. I mean, this is something that touches everybody's life. And it's such a devastating disease because the tumor sometimes grows so rapidly and we can't contain it. So what happens is there are a lot of cells in the tumor, and they keep dividing, right? They're making a new cell and a new cell and a new cell over and over and over again, and it grows. So that means you really need to make a lot of proteins to just grow, right? Because the tumor is packed full of proteins. So if you need to have a lot of proteins, guess what? You need a lot of protein factories, ribosomes. So all cancers are characterized by having upregulated formation of protein factories. And we can see that. It's sometimes even used as a diagnostics to distinguish a cancer cell from a normal cell, of how many of these protein factories they make. So the idea my colleagues and I have is that if we would just understand how those protein factories are made, then maybe we could inhibit it. And there are some slow starting points that we can see where at least, in the lab, we can maybe inhibit it, but we still have the problem that we don't understand it. It's such a black box. So we are really working hard to find the bottlenecks in this process, because those bottlenecks would be the targets for new therapeutics, so that we stop the cancer cells from making the protein factories. And our other cells would be fine, because they don't need that many proteins. They can get by for a while. We all know that chemotherapy has side effects. That kind of chemotherapy would also have side effects, no doubt about that. But it would just give us another tool in the box to fight this devastating disease. So there are two areas in RNA research that are so exciting at the moment, that some of it has made it in the popular press, and one of it has made it in the clinics, and I think the others will soon. So I thought I should talk about it, because the chances that you will hear about this sooner or later somewhere are actually pretty good. And if I can be the first to tell you, that will be my great honor. So the first one is about a story about gene scissors. So literally it is what it sounds like, how to cut genes. Why would we want to do this? Well, imagine you have a mutation that results in a disease. I gave you several examples today, right? One dream, and it's a difficult dream, and I'll talk about that later, researchers have, and patients have, and families have, is what if you could not just fix the symptoms, but cure the disease by changing the mutation. Cut out the wrong piece and put in the right piece, right? That has ethical implications, and I'll mention those later again. I just want to say that right away. But in the first place, it's technically very, very difficult, because the problem is as soon as you start cutting genes, the danger is that you cut the genome in the wrong place and you completely mess it up and everything dies. That wouldn't be too bad. But that's also the way how cancer cells arise. By cutting up DNA and putting it together in the wrong way, you get a cancer cell. So it's really, really dangerous. You need to be very precise. And for the longest time, that was just not possible, because the human genome is very, very large. So you can't just go and randomly cut it. I mean, this is insane. But in the last 5 years, so really recent in terms of research time scale, researchers found a system that works in obscure bacteria doing weird things, like nobody cared about it. And they found that this system can be used to cut genes very, very precisely. And how? It's a protein. So we have a system of a protein and an RNA. And now remember what RNA can do. RNA is a copy of DNA. And when it's made, the two can match up to each other, right? So the same thing can happen now again. The DNA opens up. I need a third arm. I always wanted a third arm. So let's imagine I have 3 arms. One is hanging over here. That's the DNA strand. We don't need that. There's another piece of DNA here. And now the RNA comes, and it can match up to the DNA very precisely because it has the complementary sequence. And by having a long stretch where the RNA and DNA match, you can make sure to find only that one spot in the entire genome and not all the others. And once the RNA has found the right DNA, and only then, the protein can cut the DNA. So we have an RNA and a protein, and they can cut DNA, gene scissors. That is a really useful tool and a really dangerous tool. What can you do with it? As I said, you can repair a mutation, but you can also insert a mutation. And in the first place, this is a really good tool for researchers because that means you can study mutations much faster. One of the most important things we need in research if we study a disease, a genetic disease, is we need model organisms, right? I mean, you don't want to go with some crazy experimental therapy and try it out in patients who are suffering already. So instead, it's very, very important to have model organisms, like a mouse model that has the same mutation as found in humans, that has similar symptoms, and then you can try out 50 or 100 drugs in the mouse first and find the 2 that are promising and only use those in patients. And making those mouse models, it used to take years, over years. It was really, really difficult. And now we can make them in 3 months, like really fast. I can't do this. This is something my colleagues are doing. But it really helps to accelerate this research. And that's a great tool. And there's so much more we can find out with it. So let's not underestimate the effect this has in the research community. But that might not be something you read about in the newspaper, right? What else can it do? Agriculture. There was a big report just very recently that you can use it to engineer tomatoes so that the tomatoes have better qualities. And I'm not a tomato expert, right? So forgive me, but I think they are more resistant to certain infections, and they ripen better. And you can use that with the system, and it doesn't leave traces of other DNA behind. So it's not a genetically modified organism. It's really just making the selection process of breeding tomato plants much, much faster. So it really helps. And I think since we are such a big agriculture community in uLethbridge, and then we have the research station here that studies a lot of those things, it is an important tool that may soon make it to uLethbridge, right? So it matters in plant research. It matters in synthetic biology. Matthew had brought that up earlier. The idea that you can use biology to make things. And that has become really powerful. You can use yeast cells to make a drug that was super expensive to make by chemistry. And now the yeast cells just make it for you. And all you need to do is harvest it. It's almost like milking a cow, right? You just take the drug, clean it up, and it's a very cheap way of making an otherwise very expensive malaria drug. And that's a huge problem because nowadays some drugs are so expensive that we can't afford them, right? It's sad. And here in Canada, we are in a good state. But if you think about other countries, affordable drugs is very important. So having the ability to make drugs in, for example, simple baker's yeast is important. But to do so, we need to change the genome of the baker's yeast. And again, these gene scissors help greatly in doing this fast. So it's fascinating how fast things are all of a sudden becoming because it's easier. I mean, it's almost like inventing a wheel, right? Life got much easier once we had a wheel. It's been a while ago. Having gene scissors makes the life much easier there. Now, the idea is out there that you could, of course, also use it eventually, maybe, to cure genetic diseases. And trust me, the researchers know that this is a difficult topic. Because which diseases do we want to cure? And which ones not? Do we want to start changing eye color, hair color? Probably not, but there's a lot of gray zone, right? There's also a question, what is a disease? What is normal? What is not normal? It's a big ethical questions. There's a lot of reservations in the field. There's also a lot of hesitation still because the technology isn't right, but often that can make a leap and a bound very quick. And I think what we're really seeing is that as a society, we don't have the mechanisms yet to debate these early enough and to make the rules early enough. And that is a problem. And I don't have the answers, but I thought it's important to mention it here because you may read about this in the newspapers. And I think you need to know both the potential and the danger and the need to regulate it. There are some applications in medicine that are very exciting. There's a clinical trial going on right now for cancer patients where it's possible, and there have been some really promising results, to take immune cells from a cancer patient, change the immune cells with these gene scissors, put them back into the same patient, and the patient agreed that he or she wanted this. That's important. You can't ask a baby, but you can ask a cancer patient. And then the immune cells attack the tumor and destroy it. And it seems to work. It's very early, but it seems to work. I think that's exciting. That might be a system where we want to use it, right? But then there are others where we don't, and we really need to find out how to define that gray zone and where to make the cut. And that's why I appreciate you all coming out here, because it starts with understanding the science to know what you can do and what you should do and what you should not do. The problem in this disease is that the muscles are wasting. So the patients are losing strength. There's some patients that have maybe learned to crawl or to walk, the children, and then they lose the ability. So instead of a child going on, learning more and more and more, they kind of go backwards. They can't walk anymore. They can't move anymore. And eventually, because of the wasting of the muscles, they can't breathe anymore. And that, of course, causes death, right? So it's really devastating. There are different severities. What I just described is one of the most severe forms that starts early. Others start later and are not quite as bad. There's a whole spectrum. And it all depends on how a certain gene is mutated. And the gene is called SMN, or survival of motor neuron, right? I mean, that says it right away. If this gene has a mutation, the motor neurons, so the neurons in your spine that control the muscles, will not survive. And that's why the muscles stop working. So you only see part of my slides, but I think that's good enough. So we'll leave it like this. Don't change that. Don't change this, please. So it will have a good ending, but this story only has a good ending because there was a lot of basic research to really understand what exactly is happening in the patients. We need that knowledge to find a cure. So let me explain to you what happens here. So there's this gene in green, SMN1, and this one is mutated in the patients. So it's not functional anymore. Usually, this gene is copied into an mRNA. That's what's shown on the bottom with the blue boxes. Now, one thing I haven't told you yet is that mRNAs actually get chopped up and put together again in smaller pieces. We call that splicing. Let's call it processing for today. Let's not make it too complicated. So basically, you just end up with the blue pieces, and all the black lines are cut out. Was that simple enough? Awesome. Thank you. I was a bit worried about this part. Oh, there we go. So the blue pieces that you saw are left over. And usually there are 8 of these blue pieces. Yes, there are 8. And that's what you need to be functional. In the patients, because of mutations, you don't get this. Okay, so that's the problem. There's one lucky coincidence here, which is applying to this disease, but not to others. And that is that this cluster of genes, where the SMN1 gene is in, accidentally, at some point in evolution, got duplicated. So there's a second one on the left side. And that one is called SMN2. It's a duplication. It's almost the same. But, I mean, usually you don't need 2 of these, right? 1 is enough. And so in evolution, that copy, that SMN2 gene, got a teeny little change. 1 building block was changed from a C to a T. That's what we show in the blue mRNA at the bottom. So that's a small change, but small changes can have dramatic consequences, because what it means is that for that SMN2 gene, when you get the mRNA and you process it, you don't get 8 boxes put together. You lose box number 7. And if box number 7 is lost, nothing works anymore. So that means this gene is also not functional. And that also means that in the patients where SMN1 is mutated, SMN2 can't rescue. Well, unless you use RNA. So, let's focus on the picture first. The top shows what happens normally. There's SMN1 gene, you put all the boxes together and you get box number 7, 6, 7, 8. Same way as you should count, right? But if you have SMN2, you skip 7. 6, 8 is not the right way of counting, so that is non-functional. But once in a while, rarely, this SMN2 gene can keep box 7. 10% of the cases, you get 6, 7, 8. So what the researchers realized once they understood this process is, if you could just push this balance, right? If we could use the SMN2 gene and push it to have this box 7 more often, that would really help the patients. So how do we push it? Here's the beauty of RNA. By doing a lot of basic research, they realized that there was a small stretch in the SMN2 gene. This is this funny ISSN1 or whatever, a sequence that inhibits using 7. So there's something that prevents 7 from getting used, and it's right in the RNA. So now here comes a scientific principle that took me long, and I'm sure you'll get it right away. It's a sequence that inhibits. If we inhibit the inhibitory sequence, that means we're not inhibiting anymore, right? If you say no, no, that means yes. Double no is yes, right? That's the same here. Inhibit inhibitory sequence, that means it works. So all we need to do is we need to inhibit that piece. Well, it's an RNA. We know how RNAs base pair up. No problem. We can find a matching piece that inhibits. And that's the red little guy at the very bottom. Small little RNA-like molecule that matches there, and then everything is fine. You get box 7 included. We call this an AON. Why do we have so many abbreviations? Sorry. It's an antisense oligonucleotide. It's a piece of RNA that matches up. It's small, it's easy to design. Of course, the researchers tested several varieties of it, but it was still relatively easy to design, and it triggers inclusion of box 7. It's easy to design, to screen, and it can be delivered. You can inject it directly into the spine of the patients. And I was very fortunate to have met the researcher who has conducted this research, Dr. Adrian Trainer from Cold Spring Harbor's laboratories, this May at a big RNA conference in the Czech Republic, where he told this story. And it has a good ending, because this RNA that he found and tested, first in the lab and then in mice, was then approved in clinical trials. He worked with a pharmaceutical company. And at the end of 2016, so really, really recently, it was approved. It's on the market. Spinraza, or nusinersen is the name they made up for it, but it really is an RNA. It will save so many lives, and it will improve the lives of so many patients and families. But what I really want to stress is it's the result of more than 15 years of basic research in his lab. Without understanding how this RNA box stitching together works, you would have never found it. And it's RNA that fixes the RNA, right? So it shows also how simple RNA is, it works. And I need to tell you one story he told us, and the story of Emma. Emma was 2 years old. Sorry, it brings on my tears, right? A 2-year-old child that was crawling and stopped crawling. She got enrolled in the clinical trial. She started getting this drug as one of the first patients. She's walking now. Can you imagine that? She's walking. It's so amazing. It's so amazing. She has visited Adrian in the lab. They're good friends. And now many more kids can do that. And it's because of RNA. And I think we need to keep going. There will be many failures, many drugs that won't work, but every one that can be found makes a huge difference. So, RNA matters, right? I hope I convinced you. Maybe not the RNA yet that we are studying in uLethbridge, but you never know where the journey goes. You need to start it and hope for the best. And there are good examples out there that RNA is a promising target and also a promising drug, as in the later example where we had both together. And I'm very grateful to be part of that. I'm very grateful that you came and listened to me. And there are many people